This is TWIS. This Week in Science, episode number 818, recorded on Wednesday, March 31st, 2021. What to do about the beasts? What should we do? I'm Dr. Kiki, and tonight on the show, we will fill your head with synthetics, rabbits, and a conservative interview. But first, thanks to our amazing Patreon sponsors for their generous support of TWIS. You can become a part of the Patreon community at patreon.com/thisweekinscience.
Disclaimer. The whole here today, gone tomorrow outlook on life can make us seize the day with the gusto of a first-year declared poetry major, evoking ancient themes of love and loss, action and change, as if all of our moments were monumental, born of some epic story never to be forgotten, meaningful, intentional, and beautiful to behold. Or we could go the other route, which is the terrible waiting between the poetic beats as we endure simply to survive. It can make us fritter and waste the hours in an offhand way. The apathy of impermanence, the fleeting worry, the fleeting joys, the fleeting terror of fleeting moments. But these aspects of life that make us aware of our brevity ignore the larger story for pages that mention us by name. This idle skimming, disjointed reading ignores the long shadows a life can cast. We are all part of a much greater story, one that has evolved over billions of years, one that was once independent, that is now dependent on our monumental choices. A modern-day world of love and loss, action and change. It is a story that can be meaningful, intentional, and beautiful to behold if only you take the time to read it. Whatever you want to do, the moment in which you can do it is always now, and where better to be now than here with us on This Week in Science. Coming up next.
I've got the kind of mind that can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening. This week in science. What's happening? This week in science.
Good science to you, Kiki and Blair.
And a good science to you too, Justin, Blair, and everyone out there. Welcome to another episode of This Week in Science. We are back again, dancing our little science booties off to the scientific beat. What a wonderful intro tonight. That disclaimer was pretty special there, Justin.
Oh.
Nice.
You mean the other ones then that I've been doing weren't
Yeah,
weren't
those
good
other
enough for you?
ones. Garbage.
I finally got one that you were like, okay, that one was good.
That one was
The
good.
other ones didn't quite hit the mark, but that one was good.
Yep.
Thank you, Kiki.
You're welcome. Congratulations. You hit my bar. Thanks for joining us tonight, everyone. On the show, I have stories about some synthetic life making great divides. Some very good stuff. And I'm theming up my stories in a conservation biodiversity bent in honor of our interview tonight. We'll be speaking with Michelle Nijhuis, who has written a book that we're going to be talking about in just a bit. But Justin, what did you bring?
That's a good question. Let me
Okay.
take a look.
Always
here it
a
is.
question.
I have jawless fish. Stuff that we can learn from jawless fish. I have a South American mystery. A socially distanced mice and hitting antimatter with lasers.
That sounds like fun.
Yeah.
Pew, pew, pew. Because that's what sounds lasers make. We all know that.
We're gonna keep
Blair, what's
fighting
in the
with
animal corner?
Oh, I have some theming today, too, that I didn't even realize. It's all kind of about evolution. So I have the origin of hopping, the origin of dreams, and the origin of venom.
I like it. It's all origin stories. You're like,
Mhm.
it's like science superhero hour. Maybe we
I'm a
should change the
regular-
name of your section. Yeah.
Yeah. Or an Aesop of sorts.
Aesop
Yeah.
of sorts. There we go. Yes.
Yeah.
All right, everyone. Before we dive into all of the science stories, I do want to remind you that if you have not yet subscribed to This Week in Science, you can find us on YouTube, on Facebook. We are on Twitch as @TWISScience, and If you go to podcast directories, we are a podcast, and you can subscribe to us on just about every podcast directory that's out there. Just look for This Week in Science. Our website is twis.org if you want to check out show notes and other details about the show. Let's dive into this science. First story of the night, I want to chat a little bit about synthetic biology. We've heard a lot for years. Craig Venter years back was like, I'm going to make synthetic life. You know, he didn't say it exactly like that, but he started a huge push to start creating synthetic organisms. And in that process, in 2016, researchers created a synthetic organism with a very minimal set of genes. It was like 470 some odd genes, a very small number of genes. And they really just want to figure out what is the minimal number of genes, what are the most basic genes that are needed to support cellular life. And so they started out with a regular bacterium, sucked out its DNA, put in some DNA that they had synthetically created. And there in 2016, their synthetic life, it was looking pretty good. It was doing life things. It was trying to divide. But when it tried to divide, it didn't do it very well. It created these little lopsided bits that wasn't evenly dividing the way that cells are supposed to divide. And so they're like, okay, we still need to work on this. Obviously, we don't have the replication part of biology down yet. And so they added a number of genes. And now they have succeeded in creating an organism that is able to self-replicate, divide evenly into evenly sized spherical cells. And all it took were 492 genes.
That's a pretty decent size.
It's a pretty decent size, but that is really small when it comes down to the number of genes that most organisms have. Most biological life has many, many more genes than just 400 or so. This research was led by researchers in the Cellular Engineering Group at the National Institute of Standards and Technology. And the hope is that they'll be able to one day be able to produce these synthetic cells to be biological sensors. And that by creating cells that can go about normal biological functions, metabolism, replication, just normal activity of cells, but then be given a special function through genes that have been added by researchers. Perhaps they can be used to detect cell environmental toxins. Perhaps they could be used to monitor temperature, light emissions from different places, and could really be used instead of engineered systems. It's cellular engineered systems. This organism is now called JCVI Syn3A. It has a very fancy name, but it's able to have normal cell division, which is a huge step.
It still seems like, see, this is what it was interesting to, that number's so big. And the reason I'm saying it's so big.
Yeah, please explain.
You have to get to a point where you need 400 genes working together in order to replicate life. So that means that before this stage of life being able to get here, say we're just going to call it, this is the smallest. They've got the right now. This is the smallest number of genes in order to replicate and have this thing live.
Right.
It means before that, you couldn't do it with 300 genes.
Yeah.
So how did you get to the 400? You couldn't get anywhere with 300 unless you had multiple organisms with a piece of the puzzle, or I don't even know if they're organisms at that point. What do you call it?
Right.
Mm-hmm. Yeah, I think ultimately you could guess, kind of based on what you're saying, Justin, that you could do it with less, but we don't know what the starting point is, right? And
that's what they're figuring out.
Engineering. Yeah. So that, in theory, now that we have the 492 gene version, you can kind of start to whittle it down and figure out exactly what is necessary and get it smaller.
And what was necessary were 7 genes to allow normal cellular division. There were 7 genes involved in that out of the 492. So it's just fascinating.
So that's a nicer, smaller number.
It's a nicer
that number.
That number, small, but it has a very discrete function. Yeah.
Yeah.
Yeah, but all of those genes, all of those molecules, all working together in tandem to allow life. Yeah. Justin, tell me your story. What's up on your first story
okay,
here?
so pop quiz.
Pop
Get rid of
quiz.
your number 2 pencils. You're not going to need them. Prepare to answer the following question in the form of an American criminal procedural comedy drama television series. Ready? What do all vertebrates have in common?
Vertebrae.
Bones. You're supposed to answer it in the form of American criminal procedural comedy
I see.
drama television type shit. Bones. Yes, they all have bones. That's the thing. Yeah, all vertebrates have bones and an internal skeleton. These bones that we have are made up of a very complex combination of composite minerals, proteins, living cells that are trapped inside the matrix of the bone. Those living cells are then interconnected by tiny channels so they can exchange electrochemical signals with each other and nutrition and allowing the bone to grow and regenerate. And there's blood production is taking place within the marrow. This is a very complex architectural system there. Where did this originate? Enter the Museum für Naturkunde Berlin team headed by Dr. Florian Witzmann. They have discovered a possible milestone in this development. They tried to go back as far as possible to look at fish that may not have had bones. They examined fossilized samples of bony armor from 2 early fish species that lived around 400 million years ago. One sample was a jawless fish that lived 423 million years ago, belongs to an extinct family. The second is much younger, about 380 million. And so they basically wanted to take a really high resolution look at this. Quoting Witzmann, it was already known that these early vertebrates had bone cells, but we knew little about how the cells were connected to each other, as well as anything about the detailed structure of the lacuna, or the cavities, in which the bone cells were located in the living animal. In order to be able to make more precise statements about bone metabolism, we had to have a far more detailed image to work from. Quick, to the electron microscopy laboratory. Hit it with the focused gallium ion beams. An electron beam scanner. Later, they had 3D images at a resolution better than any television that your eyes can handle without melting the significant part of your occipital lobe. Even in the older sample, the jawless armored fish, the 3D images display a complex network with the cavities for the bone cells and the tiny channels through the bone interconnecting these cavities. The channels are thousands of times narrower than a human hair, so this is in the fossil record that they're even finding. This is pretty amazing. And it has been almost completely preserved, they say, over the 400 million years since this thing was alive. So basically, they can see that even 400 million years ago, and these fish, the architecture was already there. It was already well underway.
That transition from cartilaginous to bony structure, right?
So even in early even early fossil bone, bone cells were able to do what they do now: dissolve themselves in places, restore bone minerals where needed. The bones themselves were acting as batteries, storing minerals, releasing them later. Our own bones do this as well. That's why they're so delicious if you're a carnivore, because there's a lot of energy stored in there.
A lot of phosphates, a lot of calcium. The bone battery, exactly. I wouldn't have thought of them that way, but yeah, they're a molecular storage device for sure. Yeah, that's cool.
Active metabolism, 400 million year old early vertebrate.
Nice. This is something that started early, and thank goodness it's continued. I am glad for that. I wonder if looking at stuff, the osteocytes, and trying to figure out how all of this stuff works. I love trying to make connections between discoveries of ancient formation and development of traits with understanding how we can approach modern medicine and possible therapeutics. So what kind of understanding at the molecular level can we get that could help something like bone disease, right? How could we do that?
Yeah, so I think one of the most powerful things that we could have would be DNA from this 400 million year old
Yes.
creature, which we
We
do
got the
not.
bones.
We
You
do
don't.
not.
Just the
We're not gonna get
bones,
it.
just the hard structure.
We're not going to get it. But by looking
That's
at something
defeatist.
like that, but trying to find its closest equivalent in the modern day ocean.
Yep.
Something that has also undergone evolution, but maybe is sort of stuck in these stages, where maybe there hasn't been a tremendous amount of change in these regions. You might see when you look at the most simplified structure, like you were talking about a minute ago, finding the most simplified structure for replication.
Mhm.
When you find the most simplified mechanisms and markers for bone growth and dissolving and everything else, you look at where our bones are now evolved to, and you can see, okay, this all has followed the same pathway. What is this that's different? Why has it changed? Is this
Why did it
improvement?
change? Yeah.
Yeah.
What forces led to that? Yeah.
And then it also narrows down the list that you might want to look at for some certain bone diseases.
For sure. Okay, Blair.
Mm-hmm.
Speaking of origins, developments, do you have one for me?
Yes, I do. And I forgot to warn you, I'm going to need you to pull up a video from this story.
Okay.
Apologies to our listeners. Please go to TWIS.org, find the show notes from today, and watch this video, because I thought I knew a lot about animals, but this is something that I had no idea about. So this was trying to find the origin of hopping behavior, where that kind of lives in the genome in animals. And to do that, some researchers from Universidad de Porto and Uppsala University worked together to study a particular kind of rabbit. So there's a rabbit called a sauteur delphore. And
sounds fancy.
It does not hop. Instead, it lifts up its back legs and it walks on its front paws.
What?
So it walks on its hands.
Yeah, it's truly wild. And I had no idea that this existed. So
And now there's a new pet that you would like.
This is some sort of weird, fake, trained rabbit meme onion story. It has to be.
It is not.
It's got powerful front paws, which you don't see on a rabbit.
Right.
And it's got these tiny little hind legs, which are usually massive.
Yeah.
Little
So,
tiny
okay.
hind legs.
So to kind of set the scene here, we know rabbits, hares, kangaroos, some rodents like kangaroo rats and kangaroo mice, their main form of locomotion is jumping. And so this paper in particular was trying to figure out why some animals move by jumping, why some animals do not, where this lives in the genome, because it turns out it's not just the powerful hind legs. It's not just, do you have the muscles for it? You have to have the quintessential building blocks. It's not something you can just build up and do. We can't build up enough muscle to just hop instead of walk. So what they did is they actually bred these weird rabbits that walk on their front legs, the sauteur delphore rabbits, with, I'm going to say, not to judge these rabbits, but normal rabbits that hop. Sorry to
rabbit shamer.
Yeah. Anyway, so they bred them together to figure out, okay, here are the ones that hop, here are the ones that don't, what's happening in their genome. So there's a mutation in the RAR-related orphan receptor B gene. So that's called the RORB gene. The RORB protein is found in regions of the rabbit's nervous system, like throughout. And this mutation that this funny sauteur delphore rabbit has a decrease in the number of neurons in the spinal cord that produce RORB. So basically, it's just a reduction in this protein. So the functional RORB gene is necessary for rabbits, and they assume other hopping animals to perform this characteristic jumping hopping gait. And that builds on previous studies in mice where they actually they inhibited the RORB gene from producing proteins, and they started walking kind of like ducks. So there's something weird related to this protein and the gait of an animal. So this shows an example of abnormal gait behavior that is mapped from a single base change. And so this is the first description of genes required for this particular locomotion. And so this points out the importance of the RORB gene and how it's part of the normal wiring of the spinal cord. So this does mean that, yes, this stops hopping in rabbits, but that also means that this could inhibit locomotion in humans or any vertebrates. It could kind of mess up how the musculature is supposed to function, essentially. So beyond this just being just a bizarre story about a bizarre rabbit that walks on its front legs and why they do that and other rabbits hop, this could actually have some really interesting implications for studies in vertebrates in general.
Absolutely. Yeah, the implications of a single base change and the effects on gait, on locomotion. I mean, yeah, this is fascinating. Why does it make a mouse walk like a duck? And why does it make a rabbit walk on its front legs instead of its back legs? This is fascinating.
Yeah.
I mean,
especially because none of the rest of the architecture should be there. It's just change gait mid-evolution. Just to, what is it like? There's certain. It's I think cows and pigs, or a lot of animals lack the thing like an attachment to the back of the skull that allows them to run fast.
Mm-hmm.
If they run fast, their head just starts going like crazy because there's no stabilizer. All your pursuit carnivores have a really nice thick thing that's holding their heads so their whole body can move and their head is still staying real focused.
Yeah.
Some animals their head just flops all over. So if this gene is messing with that channel, that's a really hard one to overcome. I imagine these have to have been a domesticated genetic fluke and not something that ever survived in the wilderness.
You would not expect to see this.
No, no.
Yeah.
Wild rabbits.
Seems like they have the musculature. Yeah, so they have the musculature to hop, but they have a neuronal kind of stopper keeping them from do that. Yeah. And so their body's like, how do I move? Oh, I guess my front legs then.
So is it
I'll just use these? I'll use what I can.
Is it just not forming the nerves all the way down? Like what is?
It's a protein deficiency.
Yeah.
Yeah.
Protein deficiency will affect it. Yeah, that'll have carry on effects.
Feedback. Yeah, but just for future reference, somebody should tell those rabbits they're doing bipedalism wrong.
Yeah, they are.
It's not the recommended way anyhow.
They could run away to the circus quite effectively. Okay. Yeah. I'm going to jump us ahead and into our interview. Hey, everyone. If you just tuned in, this is This Week in Science. And if you would like to help us grow, tell a friend about TWIS today. That would be amazing. All right. I would love to introduce our guest for the evening. Michelle Nijhuis is a project editor at The Atlantic, a contributing editor at High Country News, and an award-winning reporter whose work has been published in National Geographic and the New York Times Magazine. She's co-editor of the Science Writer's Handbook and lives in White Salmon, Washington, and she has recently published Beloved Beasts, Fighting for Life in an Age of Extinction. Welcome to the show tonight, Michelle.
Hey, thanks for having me. I love those rabbits.
Everybody wants one now.
I want one. I definitely want one.
Thanks so much for joining us. I responded to a tweet of yours where you said I have some extra galleys, and I was like, yeah, I would love to see this. And so one thing led to another, and get here, come talk to us about your book.
It was excellent serendipity, yeah.
I'd love to find out, before we dig into the book, though, you were a biologist. You call yourself a lapsed biologist. How did you go from biology to science writing and to now writing this novel?
It's a, long and tangled tale, but the short version is I majored in biology at Reed College in Portland, Oregon. And after college, supported myself by doing a lot of funny jobs helping out wildlife research projects. I was a field technician, and I worked mostly in the Southwest, so I got to, count plants after wildfires and chase frogs through streams, and most memorably follow desert tortoises around and take notes on what they ate for a diet study. And that really, I mean, the tortoises were fascinating, but what was even more interesting and memorable to me were all the politics around threatened and endangered species at the time. Desert tortoises were and still are a threatened species. And I just couldn't believe that people were talking about these profound, or fighting more commonly about these profound issues, in these meetings that were designed to, work out whether an ORV trail was going to be closed or something like that. And instead people were like, what's our responsibility to this species? Why? And I thought, wow, I know that smart people have come up with answers to these questions, but I don't know what they are. I would like to know more. So I have always been interested in the history of conservation for that reason and many others. But after I made a living for a couple of years as a field hand, I started as a journalist at High Country News in Colorado, which covers environmental issues around the Western U.S., and started writing about science, which was really my true love.
To be a biologist and then to start describing the world around you through writing, it's this transition of this studying the thing you love, and then let me tell other people about it.
That transition. That transition well. All of you do. I really like being able to. I mean, I had always liked writing, so that part wasn't so much of a switch for me. But I think I'm a generalist at heart, and I just love being able to make the connections, like you guys do every week. You make the connections between different studies and different researchers, some of whom have no idea that, what's going on in another field. And it's so cool to see those connections happen within science and then connections between scientists and the broader field.
Yeah, I love that description too, because it is the world of the generalist is getting to keep poking your head into the next lab and finding out what's going on. And if you're in any of those labs, sometimes you can be just completely siloed and so focused on either your field or specifically your own project that you don't look beyond. You don't get to poke your head in all the other doors. So.
Totally. And I've always felt vaguely guilty about it, I feel vaguely guilty about, because I know how hard science is. So I feel like, I show up, and journalism is hard too, don't get me wrong. But when I show up, at someone's field site and I'm like, well, I'm here for 24 hours. Tell me everything you've done in the last 25 years.
Yeah.
I feel a little bit badly, but I've come to think of being a generalist as a strength that the world needs. So just as much as it needs a specialist. So
yeah. On that note, though, I mean, there's always that question when people are talking about science writing and being a generalist. There's something about being able to ask the questions that an expert takes for granted.
Yeah, right. Totally.
Explain this to me. I don't know anything about it, or I know this very little bit. And
I mean, as a journalist, you get too deep into a subject also. And sometimes I'll go home to my family and I'll tell them and I found out all about this. And then they'll ask me some question that I'm like, I have no idea because I didn't ask. And it's something that's really basic, but really important. So I think we all need our perspective takers.
Mm-hmm.
So much. And so from the perspective of your studying the history of conservation and the world around you, you're kind of steeping yourself in it as a science journalist for a number of years. What led you to finally go, I have to write this book?
Well, I had been stewing about the history of conservation for a long time, always finding it really interesting and always trying to fit what my editors called too much backstory and I thought was very important context into my stories. And I just kept thinking there's a story here that at least no one I know of has really pieced together. We all know, I think, at least a couple of the famous names in conservation. People have heard of Rachel Carson. Maybe they've heard of John Muir, who founded the Sierra Club, or Aldo Leopold, who wrote a Sound County Almanac. But we don't have a sense of this is a tradition that's accomplished some things. When we think about conservation, I think, just as I'm speaking as just a typical media consumer, we're it's about endangered species. It's about these species of which there are only 2 members left. That's sort of the typical story you hear about on the news. And I knew that it was more than that. And I knew people had learned things over the last 100 years or so. And I just felt like it would be useful to put that together and kind of look at what had been accomplished, because we have done some good. We've done some good for other species in addition to doing a lot of bad. I didn't want to write a book that was just about another book about how horrible things were. I wanted to offer some kind of hope, and the version of genuine hope I felt like I could offer was, hey, look, we actually have done some other species that a lot of people don't think about, and we can keep doing them,
And I think that's really important to that, all conservationists kind of need to know what other conservationists are up to, because there's so many ways where if you have a particular species or habitat or interest that you're trying to protect, that there's overlap, right? And there's opportunity for collaboration to really amplify impact. And I think, that's something that the modern conservation movement is still, I think, trying to figure out, is how to coordinate all those kind of distinct efforts.
Sure. I mean, it's like we were just talking about, people get siloed, not only because they're super focused on their problem, but, conservation in many ways is such an emergency, and they don't feel like they can take the time often, to reach out and make connections with people who might be working on similar problems, just because everything seems so pressing, because it is pressing.
You mentioned also like a species, right? And so that's also one of the big questions is, are you saving the species where there's only 2 individuals left? Are you helping bring the California condor back from the brink of extinction? Or are you saving genes? Are you saving biodiversity? Are you saving variety?
Yeah. I mean, and I think conservationists, people who are professional conservationists and do it for a living, do have a sense at this point. We've been trying to save species from extinction and trying to preserve healthy populations of species for, on a global scale for 100 years or so. And at this point, they have a sense that conservation is about protecting relationships, relationships among organisms and among species and between species in their habitats. But I don't think the average person who cares about other creatures thinks about it that way. I think they mostly think, oh, it's about saving one species at a time.
The panda.
And about the panda. I mean, I can't, and I do have some anecdata about this, because when I was working on this book, I can't tell you the number of people who I would say, oh, well, it's about the history of conservation. They would say, oh, well, are you going to talk about the bald eagle? And I was like, well, yes. And I love bald eagles, but it's actually about much more than that. And, people who really, cared about these things and knew quite a bit about them. And I think that has to do with the way we as journalists cover those issues and the way that conservation groups present them. They always are, falling back on flagship species. And I think the media and conservation organizations need to take some more risks and, show us some more ugly species, show us some species, doing things together to show us that conservation is a much more complicated and more rewarding job than I think it is.
So we have a frequent argument, me and Blair, about what's the right path forward. I think that we should convince people to avoid going into nature, to really love heavily, overly urbanized areas with scant nature around, and give them no desire to go near nature, because even if they really love it, they can only hurt it. And I think, Blair, is your argument that if you don't expose them to enough nature, then they just don't care about it at all?
So there's some cases of ecotourism that obviously brings money and resources and awareness into areas, right? So I think I just feel like it's a little more nuanced than that, perhaps.
Really?
But what's the right solution? Because we can't keep encroaching if we are to preserve it. There's going to just be more people wanting to take the tour every year. There's going to be twice as many every 30, 40 years, whatever it is.
Yeah. I mean, I guess what I would love to see conservation organizations emphasize more is that conservation should be happening everywhere. You know, we've come to think of it as something that happens out there on the calendar picture, on the calendar landscape. And, you know, yeah, it happens there. And, you know, big reserves are really important for all kinds of reasons. But really, everyone should be a conservationist, and it shouldn't be an exceptional thing. You know, we need everyone to be thinking about it. And there are all kinds of habitats where all of us live. You know, they're not perfect, but functional. They serve other species in sometimes in really surprisingly cool ways. So I'm a little bit both and on that. I'm not.
Okay.
Yeah.
Because I mean, I'm all for getting people to be like, get familiar with your normal natural environment. Your squirrels, your raccoons, and your rats. That's basically what we're all going to be living with. That's our nature that most people will see.
Yeah, but this week there's a story that it's published in Nature that has just done an analysis and found that the average person who doesn't go out and chop down any trees, but buys coffee and products with palm oil and chocolate and these things that we love in our modern urban world, and maybe buys them without a second thought as to where they come from, are responsible for, on average, 3.9 trees being destroyed every year. And so they have found that it's people in the G7 countries, in urban developed countries, that our consumption habits are having an impact on deforestation in tropical areas that are at-risk ecosystems for biodiversity loss. And so, you know, maybe here is where the message is. You know, you don't have to be the Pacific Rim Trail hiker, right? You don't have to be that you don't have to be that nature loving, but maybe just understand that even in the city, your actions have impacts.
Yeah. Yeah, I think that's really important to understand those connections. And at the same time, I would hope that conservationists make their message more, conservation organizations make their message about more than consumer decisions.
Yeah.
Because, I mean, it's important, no doubt. Like every person who, you know, cleans up their act is saving 3.9 trees, and those 3.9 trees, you know, times however many million add up. Yeah. So they're really important, but I also think political action is important too. And so getting the word out about systemic changes that would truly make a difference. You know, the first thing that comes to mind for me is, you know, legitimizing or affirming indigenous land rights in high biodiversity countries. I mean, that's one of the most efficient and effective ways to protect
Put it back in the hands of people that were conserving for many millennia. Yeah.
Yeah. And I mean,
that could work.
Not to put the burden on them or to assume that indigenous
those skills still exist.
They're, you know, are going to just inevitably conserve their land. But just, we know from experience and research that when people have secure tenure to their land, they tend to do a really good job of protecting it for the long term. So just, I'd love to see more, you know, clean up your own act in your backyard. And yes, you know, protect your backyard from, protect the habitats in your backyard, but also think about systemic changes that can make a difference. I mean, it's also less depressing to think about systemic changes. I just have this really strong memory of seeing An Inconvenient Truth when it first came out. And I watched the whole thing, the whole slide presentation, no tears. And then I got to the end and Al Gore said, you should change your light bulbs. And that's when I welled up, because it was just so, the gap between action and problem was so huge. And so I feel like saying, you know, yes, do that. Do that on your own, do that with your household, but also plug into this larger effort, because that'll give you some constructive hope.
Yeah, and I think that also that my big problem with that whole idea also is that it takes socioeconomical and cultural factors completely away, right? Yeah, so it's kind of, it's on you to buy an electric car, to change your light bulbs, to buy sustainable coffee that costs twice as much, to do all these things. And the burden shouldn't be the financial, logistical, just the adjustment, all of that should not be on the individual. It needs to be the easy choice, right? I've brought it up a million times in the show, but it's the idea that if you have the paper towels next to the sink and the electric hand dryer behind you, you're going to grab those paper towels every single time.
Totally.
And so you need to make the correct choice, the easier one.
Right. Yeah. And that takes systemic change. I mean, I lived off the grid for many years and I had a small footprint, but then, there was no public transportation where I lived. So those efforts need to be literally plugged into a grid.
So you were off grid living. That's awesome.
Yep.
Yeah.
For 15 years?
Yeah. When I lived in rural Colorado, I lived off the grid in a house that my partner built. And, it was great. I loved it. We had solar power and we did have a very, when we were in our house, our carbon footprint was very small, but not everyone can do that, first of all. Right. And then second of all, there were a lot of things that we didn't have. We needed to share resources in order to do them efficiently, and we couldn't do that because of just the geography and
the isolation.
Yeah. You know what I mean?
Yeah.
Yeah, that's why I think those tend to work better in communities. Having a group of those that are sort of in it together, I think would be probably a little bit easier. To
share all those resources. So I was thinking about the systemic change and Blair's comment about how, it's kind of put on the consumer, and just thinking about the historical view of where these conservation efforts have been historically. The initial aspects of the conservation movement were very white male, colonial, maintaining the status quo. And you talk about that a lot in your book. A lot of the decisions and efforts that were made were to maintain that white male power imbalance. And it seems as though do you think it seems to me as though this is like a shift it to the future through capitalism and maintaining a power structure?
When we talk about individual efforts.
Yeah, individual efforts versus systemic.
Yeah, I think that's true. And the early history of conservation is full of white dude energy, unfortunately. And it has improved a great deal. And I think one of the concrete things that improvement has brought about is a greater emphasis on, community-led conservation efforts. Like I was just talking about, on indigenous and community and rural community-owned land where people really are. They might be subsistence hunters or farmers, but they really are reconnecting with their neighboring species through collective management efforts that allow them to benefit from conservation in some way and relieve the burdens and the costs of conservation. And I think those kinds of efforts do get at the kind of systemic change that's needed and get away from this, capitalism will save us. Spend more money on coffee.
And it won't alone. This is one of the interesting things, is that we have almost nobody who is involved in housing outside of some local regulatory. And so everybody who's providing housing is not providing housing. They're in business. It's all a profit business. So nobody cares really the quality of the housing outside of some sort of legal limits we've put on the minimum that can be done. It seems like if we're looking at the conservation on a larger scale, I don't see how individual communities themselves are really going to be equipped to outbid developers, outbid mineral companies, this sort of thing. It seems like we would need a sort of a federal mandate to conserve X amount of land and make it permanent. Yeah.
I mean, that's why I was talking about indigenous and rural community land rights, because, that's why it's so hard for people in developing countries to do any kind of long-term management, because they never know when some, giant corporation X is going to come in and, scrape out a gold mine And I agree with you here. There's a form of that's needed here as well. We do need traditional conservation. We do need parks and reserves because we can't let the market decide how land gets used. Conservation always costs something in the short term, and the market is set up to respond to short-term demand, not long-term, not what we all need, which is, air and water and biodiversity for the long term. I think not the long-term needs of survival. That's not what the market responds to.
We should talk to the people who are interested in the market in the terms of it's a bank. We're putting things in savings, and conservation is like putting our assets in savings, and we're gonna let them accrue interest.
Yeah.
Yeah. I think this is something we talk about on the show consistently, that there keeps being stories about this. How do we get the real economic evaluation of biodiversity and conservation reflected in the global market? How do we get that to happen? I feel like that really would jumpstart a lot of this, because no matter how bad the average person wants conservation to happen, those systemic changes aren't going to happen unless it becomes very clear that there is an economic benefit, which we know there is.
Yeah.
Science knows there is, right, but it's not trickling into that.
Yeah. And I mean, Gretchen Daly at Stanford has been working for years on ecological valuation and trying to put a price tag on ecosystem services. And, of course, there's a lot of people who say, well, you could never have the value of an ecosystem reflected in a price tag, which is true, but it would certainly be better to have it be greater than zero, which it is.
Or negative.
But here's
right.
Here's the part. Here's where I don't agree with this argument. It's partly because of bothering with that at all. It's because I don't know who that convinces. I don't think that if you're trying to influence Wall Street or government that's tied to finance, that's doing deficit spending and is doing everything short term, I don't think they like we were talking before. I don't think it even registers with them. Doesn't if they're not gaining personally immediately from it, business interests don't care what your economic numbers are throwing around. I have no interest in this. And if that's who we're trying to convince, I say we skip it. I say we just skip that whole. Put the efforts somewhere else. When you're talking about loss of a species, I mean, you look at any nations. Every state has a state bird. And it probably has a state lizard, and now they have a state flower. Yes, they probably have a state every kind of something.
They sure do.
Right?
Yeah.
Right? So we are on some very governmental level aware that we are identified by the nature in our region, and just preserving that as a state pride, or like that thing you're talking about, wanting to keep. Who cares about the turtles because we got all these trails here? Well, this is part of why people have moved there, for the nature that's there.
Right.
So you're then destroying the state itself. I don't know. I don't think the whole trying to convince the world that there's a benefit to fresh air and clean water when, every tailpipe is saying they don't care.
Yeah. I mean, I certainly don't think corporations are going to solve our problems, but I do think there's a value in there are opportunities to say, look, we've calculated that this ecosystem is worth X amount of money over the next 20 years. And, you could actually get rewarded for preserving it, for preserving that value. So there may be mechanisms that are more persuasive than what you're talking about, which I agree is not going to work. But the larger problem is that I think that conservation, because it's populated by so many biologists or people with biology background, we have worked for decades to understand what species need to survive. How much space they need, how much habitat, where it needs to be, how it needs to be protected. That's what conservation biology has done for decades. That's what ecology was doing for decades before that. But the science or the social science of how to get people to conserve land or the economics of it, all those things are just in their infancy. And so I think we just have a very rudimentary understanding of how to change corporate behavior, how to change human behavior. And, the hour is late, but there's a lot of really, I think there are so many huge questions that could be answered that, there a lot could be accomplished with just, we've been paying so little attention to it that just paying a little bit more could accomplish a lot.
Do you see?
Is that right? Is this the optimistic book that you wrote?
Yeah.
Okay. No, I'm just checking in there because. Okay. Whew.
Yeah. The optimism is we know what to do. We know what we need to do.
Yeah.
But we don't know how to get there. But we do know the questions.
Yeah.
So it's like we do and we don't.
Yeah, one of the things I thought was just fantastic about your book was the weaving of the personal stories of these personalities who were involved in advancing the field, right? In advancing conservation as a movement. The idea that because of Linnaeus coming up with a system of naming species, suddenly we could name the things and then they became important or not important, whereas before they were so vague, right? And then to move from there into, it was fascinating to me to see you weave from that initiation of the concept of a species to the Western perspective, the colonialist perspective that really was, we're going to use the land and take it over and it's ours. And I think even growing up in the 80s and 90s, I came across that perspective very often. And even into the 1990s, would see writing by people from a perspective that the land is ours. We can do what we want with it.
Yeah.
So, this is hundreds of years of time, and we're still within 20, 30 years, barely changing, barely putting a nick in that. So how much do you think, how much work, where do you think we start to put the lever against it to start to change this more?
Yeah. No question. I did try to write an optimistic book, but there's no I don't want to downplay the challenges, but I will say that, 100, when William Hornaday, who was a taxidermist and a horrible person in many, many respects, but did do a lot of good for the American plains bison and is one of the reasons why we have them today. When he decided that he didn't want to see the bison go extinct, people were really just coming to terms with the fact that humans could drive a species extinct, like that. And that was in the late 1800s. It had been known for a while that species could go extinct. And then it was kind of, Darwin had mentioned, in the mid 1800s, humans can drive species extinct, but it's okay. Don't worry about it.
Don't worry. There'll be another one.
This was like some sort of
another one. But so I guess what I'm saying,
sort of belief that overnight, each night, like the world was just populated by more animals. Like this was like a real thing that people thought. They didn't really understand breeding that took place in nature was like a process of a limited or a set number of individuals.
Yeah.
Like that concept, it really didn't exist.
Yeah, people thought that, people had a lot of faith that species were created by God and they never changed. But so I guess what I feel like it's easy to look at the history of conservation and say, well, there's been so little progress. But on the other hand, I look at it and I say, well, it was barely 100 years ago that we didn't even know it was possible to drive a species extinct. So in a way, we're way behind. And I don't want to minimize that. We're so behind, but we have learned a ton over the past century. And, we have such a sophisticated understanding of what species. And there are a lot of species on the planet today that wouldn't be here were it not for human effort. And I think it's important to remember that because it is easy to think like, oh my God, there's nothing, especially we're all stuck in our houses still because of the pandemic. It's been a hard year for optimism, but but it's like we have done good for other species. And I think it's really important not to fall into this sort of, oh my God, humans can only do damage because we've shown that's not true. It's a big list. it's possible. It's not, because we've shown it's possible, and I think it's still possible.
Yeah, I mean, there's some really amazing success stories out there. Even animals that are not doing well, and we're not necessarily our fault. There's some cases, but there are some really cool things like I think it's the Florida panther almost went extinct, but then we crossbred them with mountain lions, and then. So
they're pumas at heart, right? They're all they're all puma species.
Used our knowledge of genetics and conservation and breeding and all these sorts of things, and we were able to put all that together and save a species. And I think that it is important to stop and celebrate success because. Yeah, there's such an amazing breadth of knowledge that we've developed, and we're still reporting on every week that will help equip us if we can move forward on conservation. We have a lot of baseline knowledge that will help propel us through. And especially with like a seed vault essentially for genes. There's a lot of opportunities there with potential future use of that frozen sperm and eggs and all sorts of stuff. So
we have a lot of technical tools, and also, I mean, a lot of conservation is really pretty simple. Like most species are not yet directly endangered by climate change. They will be if we don't solve climate change, but they are. What they're still endangered by is the same thing they've been endangered by for 100 years, which is too many people are shooting them, and too many people, and too many people are destroying their habitat. And so, I mean, of course, the economics and the social science of all that is very complicated, but the facts of what we have to do are not that complex.
I have a question with respect to saving species versus ecosystems. Bringing back a mammoth could potentially benefit ecosystems. Would you be pro mammoth? What is your mammoth stance?
What is my mammoth stance? I am extremely pro mammoth in a sense, but I'm not pro mammoth in the sense that I think you mean. I feel like I'm maybe joining a conversation that's been going on here for a long time. Okay. Let's tell me what the territory is.
Justin is pro and I am essentially anti, because my thing is they went extinct due to
climate change,
systematic change on the planet, and they were no longer supported. And we have lots of other endangered or extinct species that are actually adapted to the planet we currently live on, and I would rather bring those back.
Yeah. And my point is how long is too far removed for something that may have been hunted to the brink at one point? How is the 10,000, what happens if you reintroduce the mammoth to the Canadian landscape? Is there more nutrients getting moved around in the soil? Are there different plant species that start to show up? Is that biome, can it be potentially harboring more life forms than we consider?
Could it be richer?
Reintroducing the wolf and the beaver, right, to Montana. Are there all these extra effects that we are just glossing over because,
And I stand in the middle between the two of them, and I moderate.
Yeah.
Point. Separate. Fight.
I would be as interested as anyone else in seeing a bunch of elephant mammoth hybrids running around. I'm not even going to call them mammoths because I think the whole thing is a branding exercise.
Right.
They're not mammoths. They would be. Well, first of all, they don't exist.
Yeah.
Were they to exist, they would be hybrid animals. And so we're not de-extincting anything.
So we should get rid of that Florida panther thing, because it's just See, this is how it gets. Then I get very contentious with the whole conversation.
Well,
Then we get rid of all of the hybridized conservation things.
Let me finish. I mean, my issue with it is that I don't have a problem with hybrids running around. I just have a problem with the overpromising of it. Because, and I have a problem with and this is may not particularly be anyone's fault, but it depresses me how much oxygen the whole conversation takes up. Because I feel like, conservation can be really boring. It's a gradual process, and the politics are complicated and progress is really slow. And then someone comes along and is like, you know what? We could just make more mammoths. And
it's sexy and exciting. And
I mean, no doubt the ethical issues are fascinating. The potential ecological issues are fascinating. It's fun to talk about. And then the promise of like, oh my God, we might not have to bother with all that other stuff. That would be so cool.
But do we need these sorts of ambassadors though? I mean, like sort of like what the panda has done for conservation. And Blair, I think also maybe disagrees with me on this, that the panda has done any good for conservation by taking so much of the oxygen out of the room kind of a thing. But it did get, I think, as a poster child, a way to, drum up money for conservation that could be applied to other places. I mean, we can't expect, can we, the general population to take interest in the mundane that they don't maybe understand or care about yet.
Well, I think we could try a lot harder to make the mundane interesting, and like not be so drawn to the shiny object that the so-called de-extinction that is the shiny object. And the trouble I have with it as a poster child. I mean, I think that there's some poster children have done some good for conservation. I don't think pandas have done a lot of good. In fact, there's some new research that says that actually they haven't been that great for the ecosystem.
They're trying not to exist. They're trying, as Blair is always pointing out, they're trying not to exist. They're working against themselves constantly.
There are species like that are just, you don't feel like evolution has their best interest in heart. But I feel like the other thing, of the many things that drive me crazy about the hybrid mammoth elephants, the theoretical hybrid mammoth elephant, is that it's such a primitive form of conservation. Like when 150 years ago, when we saved the bison, we put them on a train from the Bronx and trucked them out to the plains and plunked them down like they were cows. And like, that was great at the time because ecology was really young. People didn't have a sense of the ecosystem. And so basically what we're talking about is doing almost the same thing and just kind of seeing what happens to the ecosystem instead of preserving the many functioning, assemblages of species and ecosystems that we still have and that no one's doing much about.
But isn't this also like Blair's talking about this gene bank? At some point, we have to be taking on, if things keep continuing like this, we have the conservation effort, but we also then, at some point, will be transitioning into species management, where we're really controlling, like they've been doing throughout the Sierras with the certain fish population. We're up there kind of playing God with the populations, mixing up fish from different waterways just to keep the populations going and diverse enough to survive and rebound.
Yeah.
I mean, we're going in that direction anyway. I think this would be a great learning experience that would have a high profile.
Really pushing for it.
I am. I think there's angles to it that make sense in the bigger picture.
Meanwhile, there's a whole nother side of conservation that actually I would love your opinion on, Michelle, while we're talking about it, which is this idea of categorizing species as to how much effort, money, what have you, should go into their conservation. And this takes into account several things, including maybe they're too far gone, which I think is a really, I used to work at a zoo, and it was really heartbreaking when this conversation started. And I worked specifically with fishing cats. And fishing cats had been listed as a species that was maybe too far gone, and maybe they were going to stop putting money into. And, you know, I think there was a lot of controversy, and it ended up kind of falling through in the end. But I kind of would like your opinion based on the research that you've done on prioritizing species.
Yeah. Triage is what it's been called. Years. And it's tough. What actually I would love to see us put more public money into is protecting common species, because those are the cheapest species to save. And we know that when they get endangered, they're really expensive to save. And, you know, we're not giving the state governments are where conservation of common species happen. And those efforts are mostly supported by hunting. And because not as many people are hunting, they're running out of money. So we really need like a new funding model to protect abundance. But I think, you know, understandably, we also want to protect endangered species. Nobody wants to be the person to, you know, pull the metaphorical plug. I do think there's a point at which we have to say, we're not going to spend public money on this species anymore. You know, it requires too many heroic measures. It's, you know, there is a point where it's, you know, it's draining resources that would do much more good. But that's not to say that, you know, private funders can't come in and say, you know, like I spent some time at the at the Rhino Center in San Diego, where they're trying to what they call rewind the extinction of the northern white rhino subspecies by doing this, you know, very groundbreaking research on reproductive technologies, which could potentially save other species, other highly endangered species, if it's perfected. And that is mostly privately funded. And, you know, I think it's a speculative effort. They all know it's a real long shot. And I think it's valuable in its way, but I don't think we need to be spending taxpayer money on it. When that could be doing, you know, we could be getting a lot more bang for our proverbial buck.
So I guess my follow-up to that is, is there a way, can we circumvent this difficult conversation about prioritizing species by just focusing on habitats? This is like my soapbox thing, right? How can we shift? Is it possible to shift the research, the funding, and the kind of the PR focus of conservation from the species level to the habitat level, so we don't have to make these difficult decisions?
Yeah, I would love to see it happen. I think that's when, I mean, I was just talking about state government funding. That's what a lot of state wildlife agencies do, ideally. You know, some of them don't do their jobs, but that's what they're tasked with doing, is protecting habitat. And the problem we have in the US, and the problem we have worldwide, actually, with the IUCN and its red list, is that we have taken this species-focused approach to conservation. And so all of our legal structure, our political structure, and even our emotional investment is at the species level. I totally agree that would be better for the ecosystems that we want to save. I'm not sure how, politically and legally we approach it.
Isn't there a movement currently to try and get, 30% of the world's surface preserved by?
Yeah, there is. And Biden has written
all in the ocean. No.
But Biden wrote that into his climate executive order during his first couple weeks in office. And that's gained a lot of momentum. And if it gets written into the Convention on Biological Diversity, which is their 10-year goals are being set again at the end of this year, that could make, and if Indigenous land rights are written into that, which they should be, that could make a huge difference. And it could tilt, I think, a lot of the attention of the conservation movement toward habitats, which is, I think, its intent. But we've been thinking about species for a long time.
Mm-hmm.
And I think to some extent that will always be with us. But I totally agree with you that we need to be shifting the focus. And maybe talking about groups of species. Maybe we can wean ourselves from the idea by talking about communities of species and how they use certain habitats.
There's tons of ideas. People have so many ideas. And like Michelle said before, we know the science. It's changing minds. It's changing systems. It's changing patterns. This book is really fantastic. I'm at the very end of your first chapter. You end the first chapter and start the book with the paragraph. There's one more word I use sparingly here: hope. Hope is the subject of much discussion in conservation circles, both the need for it and the lack of it. Yet few, if any, of the most influential early conservationists were motivated by what might be called hope. They did it anyway. I'm scooching forward. As Leopold, in one of his grimmer moods, wrote to a friend, that the situation is hopeless should not prevent us from doing our best. And I think those are just really wonderful words. And if you were to give your message of hope and what you want people to get out of this book, what do you hope that people get from reading your book?
I hope that they can learn from this book or see from in this book that there have been more successes than many of us realize. And as we've talked about, that we do know what needs to be done. We don't always know how to accomplish it, but we know what we need to accomplish. And despite everything that's facing us, we're not as special as we think we are. that there are that a lot of people, and the reason I didn't use the word hope, as I said, was that I was just moved by all these people in conservation history who really had no particular reason to be optimistic that what they were going to do was going to work out. And in fact, most of them thought it probably wouldn't. And, they were working, at the start of the Great Depression, at the beginning of world wars, through world wars, at times of great uncertainty and chaos. And I think that they can be an example to us that they just kept doing what they thought was right and contributing in the way that they thought they could contribute the most. And I hope that people will take from the book that is still worth doing despite it all.
Thank you so much for joining us tonight for our conversation and for sharing your book. Really appreciate your time.
Thank you so much for having me. It's so fun to be here.
It's been really great having you on.
Yeah.
Yeah. So where can people find you and your book?
They can find my book anywhere books are sold. It's called Beloved Beasts. And if they want to learn more about me and what I do, they can go to belovedbeastsbook.com. That has more information about the book and where I'll be showing up over the next few weeks and giving talks and so forth.
Fantastic.
Yeah.
Thank you so much.
Hey, thank you. Thanks to all three of you. Great to be here.
It's been wonderful. Have a wonderful night.
Take care.
You too.
Bye.
All right, everybody.
I feel like this whole interview I feel like we need some kind of a call to action at the end of it.
After this whole conversation.
Yeah, yes. I feel like, it almost feels like this is, okay, I'm ready to sign up.
Read the book.
I got to read the book. I got to find out where to sign up. I got to get the book, because in the book, it'll tell me where I can be applying effort. That's the thing. I'm ready, though, now.
Oh, awesome.
But we still got to finish the show.
Ready. Let's do it. We can all apply a little pressure here and there. We can do what we think is right. This is This Week in Science. Thank you so much for joining us this evening. In a few weeks, what is it, April 17th? You will be able to find us mingling and mixing ideas, mixing our science with the technology of the Daily Tech News Show. April 17th at 4 p.m. Pacific Time, we will be science and teching it up. So put it on your calendars and stay tuned for more information as we get links and all that kind of stuff set up. And if you have ideas for what you want to hear us mind meld about with the Daily Tech News Show team, send us an email. Let us know. We'd love to hear from you. And now, as we come back from this wonderful interview, it is time for a part of the show that we all know and love as Blair's Science superhero origin story Animal Corner.
She loves our creatures, great and small. Biped, millipede, no pet at all. You want to hear about animals? She's your girl. Except for giant pandas and squirrels. There's Animal Corner.
What you got, Blair?
I was waiting. I have a really fun story. Not funny, fun story about octopuses. Always amazing when octopuses make an appearance in the Animal Corner. And this is a study looking at octopus sleep. Did you know that octopuses sleep? They do.
I did not. I mean, I guess I expect most animals to sleep in some way because that's a thing.
Yeah, and there's restorative properties to sleep, and we know that octopuses have a weird donut-shaped brain, but a brain nonetheless, which means sleep is probably important,
donuts.
right? Yes, donuts. And so this is a study all about octopus sleep. So you might have seen videos on the internet at some point of an octopus sleeping, and you might have seen this cool color and texture changing happening all over the octopus as it sleeps. And this study specifically wanted to look at if there were multiple types of sleep that an octopus exhibited, kind of like humans. So this is from the Brain Institute of the Federal University of Rio Grande do Norte in Brazil. And this was looking at, it turns out there are two alternating sleep states for octopuses, an active sleep stage and a quiet sleep stage. Scientists scientists have always thought that only humans are special. So scientists used
historically, scientists were very human-centric.
Yes, absolutely. Yes. Oh, it's so special that we do this and that. So scientists used to think that only mammals and birds had two sleep states. More recently, surprise, they found that reptiles also show non-REM and REM sleep. And a REM sleep-like state was also recently reported, kind of anecdotally, in cuttlefish. But so this brought them to look at octopuses because they exhibit this wild sleep pattern where they change their color and texture, and their suckers move and all sorts of stuff. So they captured video recordings of octopuses in the lab. During quiet sleep, they were still and quiet. They had pale skin. Their eye pupils contracted to a slit, so they don't have eyelids. So they sleep with their eyes open like fish do, but their pupil is so special compared to just normal fish eyes, because they have the ability to contract their pupil like we do. So but they have this pupil.
I think it looks like they're opening and closing their eye because the way they contract it, it's like, oh, they're shutting their eyelids. Nope, no eyelids. Just a squishing pupil. Yep. Yeah,
so their skin around their eye is changing texture and their pupil is contracting to a slit. So it looks like they're closing their eye. Yeah. So that's during their quiet sleep. During active sleep, this is when their skin color and texture were changing, their eyes moved around a bunch, and their suckers contracted, and their body twitched and moved around. And the active sleep occurs after a long, quiet sleep, which is generally longer than 6 minutes, and it has this characteristic periodicity that would repeat about every 30 to 40 minutes. Sound familiar? It's starting to sound like REM sleep, right?
Yes.
And how do they know they're asleep? Because as I mentioned, they don't have eyelids. Well, they measured their arousal threshold using visual and tactile simulation tests. It's basically they tried to wave this, more scientific than this, but it's basically like they waved their eyes in front of their hand in front of their eyes, and then they like poked them.
They're poke the octopus, see if it responds.
Hello, octopus, poke, 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 poke. Seems to be asleep. So the result of those tests showed that in both the active and quiet sleep states, they needed a stronger stimulus to evoke a behavioral response. So they were sleeping. This, of course, has implications for the evolution of sleep, because either this is convergent or there are origins of sleep dating back as far as 500 million years, because that's when the divergence between cephalopods and vertebrates occurred. So one of those two things. And so if these two different sleep states evolve twice independently in vertebrates and invertebrates, that means there's some sort of essential evolutionary pressure shaping this process in order to have a centralized nervous system. You can't decouple a centralized nervous system and sleep, is what this would mean. So if you're going to become extra smart and have a centralized nervous system, you're going to get sleep. Or, as I said, maybe everybody does it, and it has this 500 million year old connection of sleep. So the next kind of studies for this group would be to record neural data from cephalopods to understand what's going on in their donut brain while they sleep. And they'd also like to know the role of sleep in their metabolisms, thinking, and learning. So if you sleep deprive an octopus, can it kind of behave as well in behavioral tests, right? Can it perform as well? Does it affect their ability to metabolize food? Do they get tired? Do they die sooner, right? So these are all things that would be a great question to kind of follow through with. And how
necessary are these sleep cycles to an octopus's survival?
Yeah, absolutely. Does it show sleep deprivation kind of symptoms like humans do? So senior author Siddhartha Ribeiro said, I love this, it is tempting to speculate that, just like in humans, dreaming in the octopus may help to adapt to environmental challenges and promote learning. Do octopuses have nightmares? Could octopuses' dreams be inscribed on their dynamic skill patterns? Could we learn to read the dreams by quantifying these changes? So I like the idea of thinking about octopuses dreaming, having nightmares, storing memory, all the sorts of things that happens to us. But yeah, dreaming octopuses.
More study required, but it looks really interesting. And I love the evolutionary aspect of this having evolved multiple times. That,
Or not.
Or not.
That's the thing. They really kind of focus on this idea that it's coupled to brain development. But I'm sitting here going, what if like unicellular little kind of cilia covered little protists, what if they dream? We don't know they don't dream. What do you want to call dreaming? Like, do they shut down for part of the time and just chill? Is that considered sleep? Is that considered? So I don't know. I understand dreaming is supposed to be a very particular brainwave pattern and all these sorts of things. So I could be going a bit far with the single-celled organism, but I do think it's worth considering that maybe anything with a glob of nerves, anything that is kind of like a brain, could they dream? Possible.
Right. What is quiescent? What is active? What is,
This is why we need to create sentient mice and give them a dream journal so that when they wake up, they can jot down any dreams they had, and then we'll know.
But if you're creating sentient mice, then of course they have sentience, consciousness, and so then it's a moot point. Of course they're going to dream then.
Mice are sentient, guys. It's fine. They already are.
They already rule the world.
Anyway, moving from dreams to fangs, I want to talk about venom.
Okay.
Would you like to be venomous, Kiki?
Yes.
Okay, Justin? Would you like to be venomous?
I feel like at some point I'd bite my own tongue and die.
Oh, that's fair. This is
well, I mean, you'd think that if you're venomous, you're gonna not be.
I don't know. I don't know how many times
I don't think that would affect you.
I don't know how often snakes accidentally bite themselves. Yeah, I don't know how that.
It happens. It does happen sometimes because they're long and thin. You know, they're like.
And do they just like no big deal?
Yeah.
Oh, wow.
It's fine.
Yeah. Anyway, so the reason I ask is we may have found out how to make humans venomous. Let me take a step back.
What?
No,
Wait.
this is like
Repeat that before you said that fast.
This is the sensationalist headline that was all over the internet. So this is one of those stories that I'm bringing today to kind of take a step back and explain what the actual science is. So the headlines all over the internet was, how to make a human venomous. But really what the story is about is the evolutionary origin of venom in snakes, and that we share some of the building blocks with venomous snakes as mammals.
Ooh.
So, Okinawa Institute of Science and Technology Graduate University and the Australian National University have found the genetic foundation required for oral venom, and it is present in both reptiles and mammals. Yes, so there's an underlying molecular link between venom glands in snakes and the salivary glands in mammals. So instead of saliva, you could be spitting venom. So venom has arisen in so many animals and animal types. So we have venomous jellyfish, of course. We have venomous spiders, of course. We have venomous scorpions, snakes, and we have some mammals, like, for example, the platypus and the slow loris. So there are mammals that have venom. We know this already, but their venom delivery method is claw-based in their paws. So if we're talking about a venomous mammal in terms of a bite, we don't have that. So they all have different ways to deliver their venom. But this latest research wants to look at specifically the oral delivery of venom and, like I said, its connection to salivary glands. This was looking at venomous snakes, specifically a pit viper found in Asia from Taiwan. It's called the Taiwan habu snake. They looked at the genes that work alongside and interact with venom genes. They found the adjacent genes and they wanted to look at them and then compare them to other stuff. So previously, scientists have focused on genes that code for proteins that make up the mixture, but many of the toxins found in venom were incorporated after the oral venom system was already established. This is why you have antivenoms that are different for different snakes, is because their specific toxins are different. But there has to be something in common at the base of all that with the development of an oral venom system. So they wanted to look at the genes that were present before the origin of venom, which enabled the rise of these individual venom systems. So they identified around 3,000 of cooperating genes that played roles in protecting the cells from stress caused by producing lots of proteins. So they are cranking out these venom proteins, and that can cause stress to your system. They were also key in regulating protein modification and folding. If they don't fold right, they're not venomous. If they don't fold right, they could poison you. There's a million things that can happen wrong if the protein doesn't fold right. Then they looked at the genomes of other animals, and that included dogs, chimps, humans, and they found that they contained their own versions of these genes. And when they looked at salivary gland tissues within mammals, they found that the genes had similar patterns of activity to the snake venom glands. So this is the first real solid evidence for the theory that venom glands evolved from salivary glands. And while snakes then, quote, went crazy, incorporating many different toxins into their venom and increasing the number of genes involved in producing venom, mammals like shrews that produce venom, they created a much simpler venom that actually is very similar to saliva. And the ease with which the function of salivary glands can be repurposed to be venomous is surprising. It's a pretty easy switch. And so scientists could start looking at other mammals and how easily they can turn them venomous. So here's a quote. Quote, if under certain ecological conditions, mice that produce more toxic proteins in their saliva have better reproductive success, then in, just a few thousand years, you could see venomous mice.
Yikes.
Yes. So, it's probably not going to happen, but this does.
I mean, we've already got shrews. Come on.
I love it if it also, I just love it. Just the term, the snake cornering a field mouse, and then it's like the little vampire reveal.
Yeah.
The snake is like suddenly terrified.
Yes. It's like the Indiana
tables are
Jones
turned.
scene
Yeah.
where the guy pulls out a sword and then he just pulls out a gun. It's like,
And
actually,
shoots him.
yeah.
And shoots. You know how many innocent people he kills in that movie? By the way,
Well, all right,
He's
anyway.
a mass
Anyway,
murder
let's
spree.
do the show.
Let me
Anyway,
finish my
sorry.
Venom story. So obviously mice, we don't think they're on this evolutionary path to become venomous. Like they would have by now. They've been around a while. But I also like this little addition, although very unlikely. If the right ecological conditions ever existed, humans too could be venomous. It's not going to happen. There's
Not gonna happen.
There's absolutely no reason that would occur. But it's, they're just saying we have the genes. Now,
Somebody's gonna wanna.
Gene modification technology.
Somebody's gonna wanna do it.
yes.
Yeah, I mean,
Well,
you could.
You can go sci-fi with this if you want. Now, I did want to pull
Yeah,
up
sci-fi.
this story from last summer that was looking at venom glands in amphibians because I wanted to draw a connection because it's actually a pretty different story, but it's along a similar line of just venom. It's not unique to snakes. It didn't evolve a bunch of separate times. It seems like it really did have a singular origin, and then it kind of blossomed in different ways throughout evolutionary history. But this was specifically looking at embryonic analysis, and they were looking at the fact that there are oral glands in these amphibians that had the ability to develop venom. So basically, they have the same developmental embryonic origin as the venom glands in snakes, but they turn into these dental glands in caecilians, this type of amphibian. So it related, similar. So this is looking at embryonic development. And then this other story that I brought today is looking at the genetics behind it. So they could be linked, they could be related, but ultimately the origin of venom, oral venom, seems to be pretty far back, and there are vestiges of that throughout the fossil record, basically, is what this boils down to. It's in amphibians, it's in snakes, and reptiles, it's in mammals. We all have it.
Yeah. So where does evolutionarily, where does that point? Where's the convergence?
It's when good proteins go bad. It's when proteins allow an adaptation to take place, like Blair was saying. It's the proteins are there. The network of molecules that enable the protein folding and everything to happen, they're all there. And then there's a change that allows you to overstimulate your prey, or you're able to kill your prey more effectively, or like Blair was saying, better reproduction or whatever.
So we're not speculating then that there was some ancient ancestor that we're all linked to that had venom for a little while and then just dropped it. It
seems possible. So I think this is the
Maybe.
thing that future research is going to have to look at. Yeah. But based on this story we brought last summer, that there's the developmental origin of what gives us venomous snakes is also in these caecilians, these amphibians.
Amphibians, yeah.
Would lead you to believe that the common ancestor between reptiles and amphibians had at least the developmental building blocks for this. But that's the thing where I think it gets really difficult, is that in the caecilians, it developed into these weird, like, leaky dental spoon-shaped things in their mouths, and in snakes, it turned into fangs with injectors. So did their common ancestor also have venom capabilities, or did their common ancestor just have the developmental possibility to create venom, right? And so I think that is where it becomes really difficult, is the genes are there, the structures are there, but did they develop to completion?
We don't know.
However many millions of years ago. And that is what we don't know.
Maybe it didn't, and that's why they didn't stick around.
Right?
No.
Right?
Quick
Yeah.
shout out to Dan Christensen and I think it's the YouTube chat room for the in-chat donation and for correctly calling that. Yeah, you're right. Shrews are venomous.
Yeah.
That's
Mhm.
Called it ahead of the reveal. Or that part of the story.
That's
the
right.
head of the reveal. You know what I'm going to be revealing right now?
What?
That Justin, it's your turn to talk about science.
Ooh.
Science. Okay.
What story are you
this
gonna
is
hop
an interesting
on?
story. This is a team of researchers from the Universidad de Sao Paulo, Universidad Federal do Rio Grande do Sul, and Universitat Pompeu Fabra, which I think are all in South America somewhere. Anyway, they found evidence of genetic Australasian influence in more parts of South America. If you weren't aware that they had discovered any genetic early Australasian influence in South America, apparently there was. I didn't even know about this study. Somehow I missed it as well. Back in 2015, researchers found what they described as Australasian influence in native people living in the Amazon. That would be people with genetic markers associated with the early populations of South Asia, Australia, and Melanesia. In their papers published in Proceedings of the National Academy of Sciences, the group now describes their study, this next round study, of a genomic data set from multiple South American populations across the continent that are also showing this sign. So, theories that explain how the signal could have been introduced into people living in South America are tough, considering it has not been found in any early people living in North America. Current population theory is that North America and South America were populated by people migrating over from Asia via Siberia to Alaska, then waiting for a glacial event to melt, and then continuing down and then populating North America and then South America, although they might have kind of headed to South America along the coast quicker. Sort of tough because while this is upheld by most of the evidence we have, it isn't perfect. The oldest human activity that we've seen in the Americas is now this cave in Mexico that's 30,000 years old. There are a number of South American sites that are likely somewhere between that 30,000-year-old age and then the oldest stuff in North America. So if you were just going to go off of no other information but the age of the site, you would say started in Mexico, headed to South America, and then moved to North America. Now, the genetic information still is backing up the transition from Siberia to the Americas. That's still the correct overriding theory.
Yeah.
But this experiment that they just did involved collecting blood samples from native peoples all across the midsection of South American continent. They conducted the genetic analysis, and all they studied samples from 383 people, included 438,000 plus markers within the genome that they were looking for. Researchers found the Australasian marker in native people living in the Brazilian plateau, in the center of the country, and also those living in the western part of the country. They also found the signal from the Chotuna people in Peru. Findings suggest migrations of people were far more widespread in South America than were thought. The findings also suggest that there were a couple waves of such migration. This has led to some scrutiny of previous theories about how people got to South America. Some suggest, though, that this may be a problem because many lineages in North America were wiped out by European colonial genocide. So we might not have a full picture of North American genome, and I don't know how much access we have to ancient North American genome because of some treaty issues that have taken place where we're not really allowed to do a lot of analysis without a lot of express permission, and that permission has been begrudging over time to look at ancient populations.
Yeah.
We have had some exceptions, but it's, for the most part, we don't have a very good map of North American indigenous.
Fascinating.
Yeah, so closer study, though, they also think, though, there's another thing that says, if we just look at that North American population, the data that we do have, we should eventually find this signal there as well.
Yeah.
Most enticing possibility is, of course, that a population of early people from Australasia somehow made their way all the way across the Pacific directly to the shores of South America, which is not supported by any other evidence at this point.
Aside from the fact
Aside
the
from a
genetic
marker.
the genetic marker. Yeah.
Yeah, which is pretty good sign. That's a pretty good signal. But it can't be ruled out either because of that genetic marker. So yeah, like there's a lot of interesting questions that arise from this.
I mean, people have rowed across the Indian Ocean, the Pacific. I mean,
that's the other thing about it too, which is it wouldn't need to be that big of a population.
Yeah, it's a long trip, though.
It's a long trip, but it's also not. It's not saying that the Americas now were populated by a cross Pacific, Southern Pacific crossing event. Mm-hmm. It's not there, but the admixture does tell you that either when people showed up to South America, they might have run into people who were already there, or, you know, could have been the other way. We don't know. We have no idea, but it's this absolutely fascinating question, puzzle thing that needs to get tracked down. Very exciting. This is a very exciting time for paleontology.
So cool. Need more data.
More data.
We need more data.
Oh, and then more stories. More stories.
You have more stories.
Yeah.
Cool.
So this is quotey voice of Gloria Choi, Associate Professor of Brain and Cognitive Sciences at MIT, member of the Pick-Our Institute, if you're familiar. Quoting, as a community, it's very important for animals to be able to socially distance themselves from sick individuals, as all of us now totally understand. Especially in species like mice, although I'd say humans, safe bet too. Especially in species like mice, where mating is instinctively driven, it's imperative to be able to have a mechanism that can shut it down when the risk is high. To explore whether mice would change their innate behavior when exposed to sick animals, researchers placed mice in the same cage with either a healthy female, that's male mice, must be. Yes, male mice in the same cage with either a healthy female or a female that was showing signs of illness. And they had this bacteria that they injected with them that kind of made them sick, but wasn't. They found out that the males engaged much less with the sick females and made no effort to mount them. So the whole got a headache thing, apparently. I think you have to go with fever. I've got fever and cold sweats, and I have an off pallor. Anyway,
you don't look so hot. Why don't you stay home?
Think about just having like drip it, just like so congested, just like the bright red nose and the
not a problem.
The covered in just tissues in every pocket. It's just really just maybe doesn't get you going, and that might be why.
Right. Yeah. Doesn't really. Those aren't really the like I want to date right now. So, right.
Yeah,
no.
exactly.
Snot, vomit. Even
there's a reason. There's a reason that being sick is like quote unquote gross, right?
But what is that reason? Researchers.
It's a go away signal.
But what is that signal? Researchers didn't stop with just that observation. They tried to identify the brain circuit underlying the mouse overcoming their instinctual behavior of wanting to mount the member of the opposite sex. Mice have a lot of intercourse. It's we talk about bonobos a lot. Mice, big time into the sex thing. Okay, so what they said the Bemmer, I'm gonna have to phonetically, I'm sorry. Vomeronasal organ, which processes pheromones, feeds into part of the amygdala called the coa-PM, coa-PM, and the MIT team found that this region is activated by the presence of the bacteria-injected mice. Further experiments revealed that this activity of the coa-PM is necessary to suppress the male's mating behavior in the presence of sick females. when CoAPM activity was turned off, males would continue to try to mount sick females. Wasn't the appearance of snot. They needed to have this signal in the brain, which is probably being triggered by that.
There's a trigger.
There's a trigger. Additionally, artificially stimulating the CoAP suppressed mating behavior in males even when they were around healthy females. So they could actually stimulate this and get them to, I guess, think everybody was sick or act like everyone else was sick.
Yep.
The research also showed that it communicates with another part of the amygdala called the medial amygdala, and this communication, carried by a hormone, is necessary to suppress the mating behavior. The link to this which is called thyrotropin releasing hormone, TRH, is intriguing, Troy says, because thyroid dysfunction in humans has been implicated in depression and social withdrawal.
Yeah, that's an interesting point on this study where it's thyroid dysfunction could have this influence on human behavior, not the mounting effect, but just like you said, social withdrawal, people choosing to not be around other people more often because there's a signal in their brain that is kind of pushing them to do that.
But what's really interesting then is it sort of changes your perspective of why somebody is being a little antisocial and depressed in this sort of way or withdrawing. If they're having the same feeling that Blair would have if everybody was walking around with number 11s, right, coming out of their noses, then yeah, all of us would be like, I'm really not into hanging out with you people today because it's me. I just don't feel like it. Not because you're all revolting.
Yeah. I mean, it's such an interesting question where there's a behavior when you look at it in a model species like a mouse that is this repetitive stereotyped behavior that, a mouse is going to do it, no matter what, unless you take that part of the brain and go, you're not going to do it anymore. Right. And so you start seeing that there are these levers in the brain, these switches that can be turned on and turned off that lead to different behaviors. But then when it comes to an animal like humans that are a bit more complicated than a mouse, we like to hope, that because of the layers of cognition that are involved in certain pro-social behaviors, they are going to have effects, but maybe not the same effects as they would have on a mouse, right? But it's really interesting to dig into this on an animal level.
I think it also is interesting to think about it on a system level because one of the things I just thought of too is that, not to bring it to humans again, but the human flu season is also the stay at home and nest season. And, we always assumed that was just because it's cold out, but maybe there's something else going on there.
Right. We're seeing people sick and going, I'm just going to stay home. I don't need to be around other people right now as much. Yeah.
Mm-hmm.
Could be bigger.
I'll do a very short version of the story. Researchers with the CERN-based Alpha collaboration have announced the world's first laser-based manipulation of antimatter. Leveraging a made-in-Canada laser system to cool a sample of antimatter down to near absolute zero. This is published in the journal Nature, and it is considered an absolute breakthrough because they used a made-in-Canada laser.
Justin.
Incredible. Didn't think they were ever going to get that done. Wow. My shortest story.
That's your story. That's your take on the story.
I can keep going with the story. That's, to me, there's stuff about, being able to then, now that you can control a little bit of antimatter, now what research can we do? This is something that they've been working on for 40 years.
Yeah.
It's quite a breakthrough, but really the highlight of the story, as you can tell from the fact that it is in the first sentence of the press release, is that it was done with a made in Canada laser. They're very proud.
They're very proud.
We did it. We made a laser. It's just, amplifying light down a tube. It's very, very impressive.
It is amplifying light down a tube, and that these lasers are helping them manipulate antimatter, which we're just getting a handle on. This is very exciting for our understanding of the universe. Thanks, Canada. Thanks for your lasers. We appreciate that. All right. I have some stories. First, I want to say thank you once again for joining us for This Week in Science. All right. My science. What do I have?
Wait, sorry. This is just coming in. Okay.
What?
It was assembled in Canada, I think is.
Made in, assembled in. Oh, verbs. Oh, verbs.
Sorry. I couldn't resist. Go ahead. I'm sorry. I'll stop being the interrupting cow.
Okay. All right. So I'm going to start with a story that is a follow-on from the interview and from conversations we have also had on the show many times about invasive species and conservation. Well, it turns out that snakes, rats, cats, all the invasive species in the world, according to researchers in France, that invasive species have cost $1.3 trillion to the global economy since 1970. It's an average of $26.8 billion per year. And it's almost as much as, it's not quite as much as the infrastructure bill that's being proposed right now. But anyway, study published in Nature, researchers have been counting all the costs that invasive species have had in the world. And they came up with a top 10 number of invasive pest species. They're groups of species, mostly, that are having the most influence and doing the most damage around the world. And on this list, I named a few off the top, but let's talk about who the most costly invasive species are. Mosquitoes. They are by far the most costly invasive species at a level of $148.7 billion. And then we have rats and mice coming in at 67.1. We have cats. Cats are number 3 on the most bad list of invasive species.
I'm only surprised they weren't higher.
I know, they're third. And then we have Formosan subterranean termites, red imported fire ants, brown tree snakes, Mexican cotton boll weevils, gypsy moths, Africanized bees, and New World screwworms.
Screwworms.
That's right. They screwed us. Anyway, there's a lot for these animals and the trouble that they have been causing. And guess what?
What?
Another study has determined that the pets that we like the most, hey cats, what's up, are most likely to become invasive species. That there is a certain combination of traits that leads to basically generalists that are able to adapt to ecosystems and be able to survive. And they did some mathematical modeling and then some experiments with ants, and have pretty much come to the conclusion that we're bad people for the pets that we like to choose, because our pets are highly likely to become invasive species that are then going to go on and cost trillions of dollars to the global economy.
Well, they probably started it, right, as an invasive species to begin with. But they were so adaptable. They were already out serial killing. They were adapting to anything.
And then we said, hey, be our pets. No.
But they got, I think so. They probably just were amenable enough to be around people. Like, we were this close to, like, if raccoons and squirrels could just chill out a little bit more and play along, they could take the role of cats and dogs. They could be like, Are you a cat person or a dog person? Or a squirrel person or a raccoon person? you'd have to ask all these questions. But they never made that transition quite. But the dogs and cats probably just hung around being invasive with humans.
So that's usually not the direction it goes. Usually we decide we like pets, and then the pets get into the environment and start causing trouble. And the one thing about this study that I thought was really interesting, their analysis, they started out looking at which species are more or less likely to be invasive. And if pets were not more, if pets didn't have any invasive characteristics to them, if they're not likely or not at all or whatever, if they're just not that invasive, you'd expect that they would be equally distributed among all the species. But what they found is that invasive mammal species were present at 5 times the rate in the pet trade as they are in the wild around the planet. So it's similar for birds, for amphibians, and all sorts of different animals, fish. Anyway, all the species, we like invasive ones as pets. We probably made them invasive, but anyhow.
Well, there are places where they purposefully released cats too. Cats like when I lived in Israel, there were
to kill the rats, which were invasive.
Yes, exactly. Yes. So the Brits were like, "Oh yeah, let's just release a bunch of cats out there to take care of the rats and mice that we accidentally brought here."
Yep. And then I don't know. On a good note, though, if you do happen to have a garden and you want to do something good for the world, know that your urban garden is having a beneficial effect on pollinators. A new study that just came out in the Journal of Ecology suggests that urban areas in the United Kingdom, this was in the UK, are a significant source of nectar and floral diversity for insects. And so this is a good thing. They were looking to understand how the nectar supply differs between urban and rural areas and where nectar sources are concentrated in urban settings. And so they started looking at where things were. And basically they discovered that your garden can be a refuge and a source of sustenance for those pollinators that we need.
And the reason that they're pointing to your garden is because everywhere else that is controlled by the city, they're not going to take the effort to maintain something like wildflowers. They're going to put crushed granite and concrete over everything.
Possibly,
so there's no there's
less lower maintenance. Yeah,
there's very little. This is sort of also the thing I was thinking about when we were doing the interview. There's very little land where I'm at anywhere that is not heavily managed by humans. I mean, granted, most of my surroundings are farmland, which are by nature completely manipulated by man. But within the city, there's not really any wild areas.
So listen to this, though. Listen to this, though. So this is in the UK. The researchers sampled in and around 12 towns and cities. They were in areas categorized as urban, farmland, or nature reserve. And urban and farmland had higher numbers of non-native plants than nature preserves. They found that all 3 of them had about the same amounts of nectar. The urban sources of nectar, 85% was attributed to gardens. However, even paved areas contributed to nectar sources, in some instances with flowering shrubs growing through cracks or in parking lots. But within gardens, 83% of nectar-producing plants were non-native. So
that's always a problem.
That's an issue also, but there's nectar, and that's good for the insects.
This is where Justin's green belt he was talking about before would be super helpful.
Oh, yeah. Yeah.
Yeah, green space is so important. If you can do it in your backyard, but kind of part of the community level thing we were talking about with our interview is if there's legislation or particular representatives who want to favor more green space in your area, that is another really important thing to support. Yeah. Because that's ultimately you need kind of that higher level support to make sure that land is saved or converted back to green space when possible.
Yeah. Yeah, possibly. Okay. I'm going to take us away from conservation, though, and now I'm going to take us in robotic sci-fi land. It's here.
Great.
It's here. Last year, researchers reported their Xenobot. The Xenobot was a biological robot created out of layers of skin cells and cardiac cells from Xenopus frog embryos. So they called it Xenobot. They created these little micro machines out of biological parts that could function, that could move around. And they moved around and they did little things. And they're like, look, biological robot. This is so cool. Well, they went back to the drawing board and decided to play with their design of Xenobot a bit further. And now they have the sequel to Xenobot and have created, as they say, a cellular platform for the development of synthetic living machines. They are reporting a method for generation of in vitro biological robots from Xenopus laevis cells, so frog cells. They exhibit coordinated locomotion via cilia present on their surfaces. So they took frog embryos, took stem cells from the frog embryos, allowed those stem cells to grow into little spheroids, little balls of cells, and those little spheroids then started to grow little hairy appendages all over them. Cilia, naturally. It's a natural process. It wasn't something that the researchers made the cells do. The cells did it themselves. There are lots of natural, normal processes in which cilia arise in biological systems all the time, like our gut lining and all sorts of things. So these little cells got cilia all over the outside of them. And so the researchers took them at that point and they started using them to make little robots. And so they basically have created these little robots that are amenable to high throughput projects. They arise by this cellular self-organization. They don't need any external stuff, no scaffolds, no microprinting. And you can also, they can be damaged. They tested them. They cut these cells, like a significant slice out of the cells, and the cells healed themselves within 5 minutes and were able to go on about their business. They're amenable to surgical, genetic, chemical, and optical stimulation during this self-assembly process. They can navigate aqueous environments in diverse ways. And they have emergent group behavior. So these little tiny cells, there's a video of these little tiny cells wiggling around. And in my view, I don't think it looks emergent. Group behavior doesn't look like, but they are moving around in the same area together. And in their motion are able to potentially collect little iron particles in the environment and move them out of the environment. These are not smart machines, but the machines have been, they have a genetic switch, a genetic trigger to have a memory of 1 bit on or off. And so with this genetic trigger, which is a fluorescent protein, if they are exposed to blue light, they will fluoresce and they will have that memory of, quote unquote, memory of having been exposed to a particular light signal. So what they can do with this kind of model system is send these little frog bots into environmental, biological ecosystems, into areas where they can play cleanup. They're thinking about using these Xenobots for cleaning up microplastics, and all sorts of things that are difficult for humans to do, but that these little frog bots kind of do on their own. Frog bots, Xenobots.
Spent so much time wondering if you could. Didn't stop to think about if you should. Yeah, that's interesting. Kind of scary.
it seems as though it's pushing this single, because these are single cells. These are little cilia covered single celled frog organism bots. So yeah, is it pushing things in an evolutionary direction? Would they be influenced in some way? They are biological. They can adapt. If they survive and reproduce, then that potentially becomes something. At this point in time, they survive a short period of time as long as they've got amniotic fluid nutrients to be able to rely on. Otherwise, they go into a sort of stasis if they are in and can remain in a stasis state for a fairly long period of time if they run out of nutrients or if they're taken out of a nutrient situation. But they can't reproduce on their own. They're just frog cells that
So
move around.
on one hand, essentially remote controlled single-celled organisms could be super helpful in the medical field.
Yes.
Super helpful. On the other hand, could kind of go the other way.
On the other hand,
It's one of those technology falls into the wrong hands sort of thing.
yeah.
With the great medical and military applications of our new technology, we will save half and kill half of the planet at any time we choose.
That's right. Snap. It's a snap.
Yeah, it seems always to be there also this sort of deficit anytime we have done something that is very designed in terms of life, which is that we have created something that can perform a task that maybe we would not have been able to just find anything in nature to simulate the activity. We can really engineer that. Only problem is we don't have all of the rest of evolution that took place to make a thing able to survive stochastically against many situations. So we're creating these born sick hybrid poodle things that are really good at fetching. But then, if the temperature goes up 5 or down 5 degrees, they just die. They just can't
exactly.
survive.
I mean, at some point, maybe there will be these biological soft-bodied machines that we'll be using, that will be helping us. Right now, what this is a platform for investigation. I mean, there is an emergent swarm behavior in these little single-celled organisms. That's interesting. Maybe there's something to learn there about swarm behavior. So the bioengineering aspect of it also is, that's still under development. This is definitely not released the frog cells into the wild stage yet at all. But Xenobots 2, the sequel, has arisen. It has.
Stuff does escape from labs. Where's it I don't know where it is, which Jackson lab it is, but they had the escape, I think. Maybe not, no, it was probably not at Jackson, but somewhere they had the Doogie Howser mice escape from a lab. The Doogie Howser mice.
The rats of Nim.
Yeah, the Doogie Howser mice were these mice that were, are they rats? Maybe they're rats.
Yeah, they were mice. Yeah.
They were designed to be exceedingly intelligent compared to other mice. And they were being utilized for experiment because they had apparently really good memories and some decent, cognitive abilities. So they were really fun to run these complicated cognitive experiments on. And some of them have escaped over the years and out there in the wild breeding a potentially super intelligent race of mice that is interesting that we haven't heard anything about
in a while.
Either
that or they bred with wild mice and bred the smart right out of them.
Mm-hmm.
You have to.
My money's on that one. For sure.
Yeah.
You have to, at some level, always acknowledge when you're dealing with anything that you're going to let it out.
Yep.
Inadvertently, of course. Accidentally, of course. Unknowingly. This is always true with every chemical compound that we create. Just like every drug has a side effect that we don't find out about until 20 years later, 30 years later, 40 years later.
Just like my anxieties and my stresses, I just push them deep down inside, but eventually they come out. No.
Like so much microplastic.
Like so much.
Your bodily fluids. Yeah, it's going to happen.
It's going to happen. But you know what else is going to happen?
What?
We're going to get to the end of the show.
Oh.
We did it. We did get to the end of the show.
Did we do the shout out for the get together thingy?
I did it once, but I will do it again now.
I missed it. It's my fault.
Yeah, you left the room. But a reminder, everyone, put it on your calendar, April 17th for the TWIS DTNS Crossover Science and Technology Show. 4 p.m. Pacific Time, April 17th. Mark your calendar. It's going to be great. And let us know what you want us to talk about. You really should. Okay, so now it's time for us to say the thanks and the things and there's my run sheet. It's time for a few shout outs. Shout outs to Fada. Thank you for your help making the show notes and the descriptions, the social media, doing all that work. Really appreciate it. Gord, thank you for all your help in the chat room. Identity 4, thank you for recording the show. And Rachel, we know you're real. Thank you for your assistance. I would also love to thank our Patreon sponsors for their continued support. Thank you to Karen Tozzi, Woody MS, Andre Bissett, Chris Wozniak, Dave Bunn, Vegard Shefstad, Hal Snyder, Jonathan Stiles, a.k.a. Don Stylo, John Scioli, Guillaume, John Lee, Ali Coffin, Gaurav Sharma, Shubru, Darwin Hannon, Donald Mundus, Stephen Aldebaran, Daryl Myshak, Stu Pollock, Andrew Swanson, Fred S104, Sky, Luke, Paul Ronovich, Kevin Reardon, Noodles Jack, Brian Carrington, Matt Bass, Joshua Fury, Sean and Nina Lamb, John McKee, Greg Riley, Mark Hessenflo, Gene Tellier, Steve Leisman, a.k.a. Zima, Ken Hayes, Howard Tan, Christopher Rappin, Dana Pearson, Richard, Brendan Minish, Johnny Gridley, Kevin Railsback, Flying Out, Christopher Dreyer, Mark Massaros, Artyom, Greg Briggs, John Atwood, Robert Coburn, Rudy Garcia, David Wilkinson, Rodney Lewis, Paul, Rick Ramus, Matt Sutter, Philip Shane, Kurt Larson, Craig Landon, Mountain Sloth, Jim Drapeau, Sarah Chavez, Sue Dostard, Jason Olds, David Neighbor, Eric Knapp, E.O., Kevin Parachan, Aaron Luthen, Steve DeBell, Bob Calder, Marjorie, Paul Stanton, Paul Disney, Patrick Pecoraro, Tony Steele, Ulysses Adkins, Brian Condren, and Jason Roberts. Thank you for all of your support on Patreon. And if any of you are interested in helping us out on Patreon, head over to twist.org and click on that Patreon link. And on next week's show, it's World Beaver Day next Wednesday. And we are going to have an interview with a beaver expert next week.
No way, really?
Yes, way. Yes, it's very exciting.
So yeah, on next week's show, we will be back Wednesday, 8 p.m. Pacific Time, broadcasting live from our YouTube and Facebook type channels, as well as from twist.org/live.
Want to listen to us as a podcast? Just search for This Week in Science wherever podcasts are found. If you enjoyed the show, get your friends to subscribe as well.
For more information on anything you've heard here today, show notes and links to stories will be available on our website, www.twist.org, where you can also sign up for the newsletter.
You can also contact us directly. Email Kirsten at thisweekinscience.com, Justin at twistminion@gmail.com, or me, Blair, at blairbaz@twist.org. Just be sure to put TWIS, TWIS, in that subject line, or your email is going to get spam filtered all the way into the dreams of Octopus.
Wow, that sounds really complicated. You know what's even better and easier? Hit us up on the Twitter, where we are at Twist Science, at Dr. Kiki, at Jacksonfly, and at Blair's Menagerie. We love your feedback. If there's a topic you would like us to cover or address, a suggestion for an interview, a haiku that comes to you in the night, please let us know.
We'll be back here next week, and we hope you'll join us again for more great science news.
And if you've learned anything from the show, remember,
it's all in your head.
This Week in Science. This Week in Science. This Week in Science. This Week in Science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And oh, it'll is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth, and I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 I've got one disclaimer, and it shouldn't be news, that what I say may not represent your views, but I've done the calculations and I've got a plan. If you listen to the science, you may just yet understand that we're not trying to threaten your philosophy. We're just trying to save the world from jeopardy. 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 And This Week in Science is coming your way. So everybody listen to everything we say, and if you use our method instead of rolling a die, we may rid the world of toxoplasma gondii. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 Got a laundry list of items I want to address, from stopping global hunger to dredging Loch Ness. I'm trying to promote more rational thought, and I'll try to answer any question you've got. So how can I ever see the changes I seek when I can only set up shop 1 hour a week? This Week in Science is coming your way. You better just listen to what we say, and if you've learned anything from the words that we've said, then please just remember it's all in your head. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 This Week in 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 Science. This Week